Welcome everybody to the Roslin Institute. Please take a seat. So it's a very welcome to everyone at this very special occasion that sees the inaugural lecture of Professor Elspeth Milne, who is the Professor of Veterinary Clinical Pathology. Now, as Elspeth I'm sure will attest to, it's been a little while since she was actually appointed, actually one of the longer ones, about 9.5 years ago. So we've finally got there. For those of you who don't know me, I'm Mike Shipson. I'm the Dean of Biomedical Sciences. And as probably many of you realise, that normally these prestigious occasions are convened by the head of college. Unfortunately, our interim head of college, David Argyle, is on business in Denmark. So Elspeth, apologies and apologies to everybody else. So today you've got the stand-in for the interim head of college. So welcome colleagues and friends, in particular to Elspeth's husband Fraser and her brother Richard. I'd particularly like to make a very special welcome to all the visiting pupils from the following schools that we have today. From Beeslack Community High School, from Firhill High School, James Gillespie High School, Laswade High School Centre, Queensferry High School, St Margaret's Academy, and the Royal High School. And I think especially for the pupils in the audience, Elspeth's lecture is going to be of particular interest because I'm sure she's going to highlight opportunities that a veterinary career can provide and really indicating the extensive range and real diversity of things that you can get involved in as a veterinarian. She's going to be talking from cell biology right through to conservation. Now, Elspeth started her veterinary training here in Edinburgh with her Bachelor of Veterinary Medicine and Surgery, and she also completed her PhD in Edinburgh. After a short while away, she now currently heads the Easterbush Pathology facility, providing a very extensive diagnostic and clinical pathology service to all on this site. Elspeth is an RCVS recognised specialist in veterinary clinical pathology and is also a fellow of the Royal College of Pathologists. With no further ado, I'm going to invite Elspeth to give her inaugural lecture, which is entitled From Cells to Clydesdales, a romp around the veterinary profession. Elspeth.
Thank you very much. Sir John, Dean of Biosciences, colleagues, school pupils, and members of the public, I'd like to thank you very much for coming to attend my lecture. It's nice and encouraging for me to see such a good turnout. So, as the Dean said, it's entitled From Cells to Clydesdales, a romp around the veterinary profession. I did notice in some of the original flyers that went out for this, and actually some of the recent ones too, that there were some words missing. And I thought I might be the first person in the history of the University of Edinburgh to have their inaugural title censored. But I'm assured that it was just a mistake. The reason for using those particular words was that I wanted to convey that while veterinary medicine is a serious business and sometimes even a harrowing one, you can have a lot of fun, and I certainly have. So when I was trying to decide about 6 months ago what I was going to talk about, I realised that my career had been so varied that just to pick out one particular thing and talk about that in great detail didn't really do justice to it. It wouldn't be very meaningful. So instead, I decided to make it a celebration of the variety of jobs that one can do as a veterinary surgeon with a veterinary degree. It seems traditional just to talk a little about one's journey to becoming a professor. At least in geographical terms, my journey has been about as short as it could possibly have been. Here we are at Easterbush here. For the first 8 years of my life, our family lived in Poulton on the side of the valley of the River North Esk. And when I was 8, we went all the way down the hill and all the way up the other side and moved to the edge of Lone Head. And these houses are actually looking over at each other. So the North Esk Valley played a large part in my early life, and I walked, climbed, rode horses, and generally knew that area like the back of my hand, and it's not really changed that much today. So initially I went to school at Laswade, down just off the map here, and then subsequently to George Watson's College in Edinburgh. Just to talk just very briefly about my parents. My mother was, although she came from the centre of Dundee, was for some reason decided that she was going to do agriculture. So she went to the University of Aberdeen and graduated from there in 1948. My father was a geneticist and developmental biologist, and he worked at what was at that time the Institute for Animal Genetics in Edinburgh at King's Buildings. And he performed the first successful non-surgical transfer of mammalian eggs. And he received the Royal Society of Edinburgh Award, a Keith Medal, in 1962. He was also, although sadly he wasn't around to know this, but he was also PhD supervisor of Robert Edwards, who got the Nobel Prize in 2010 for his work on in vitro fertilization in humans. And it was very nice that my father was included in the Towards Dolly exhibition that the university had a couple of years ago as well. So, in the beginning, the usual pictures of one aged 5 or something. I think in this case I was about 6 on the left-hand side holding a wild eastern snapping turtle when my father went to do a sabbatical in Cape Cod in Massachusetts, and we all went with him, which was very enjoyable. But as a child, I was obviously extremely interested in animals and all things natural history, as well as domestic. And here, holding up a fish that I've got, seemed to have been keen to hold up things to the camera in those days. So on the basis that if you cross a scientist with an agric, you're probably going to end up with a vet or something similar that's interested in large animals. And so it turned out. So in 1979, I graduated from the Royal Dick School of Veterinary Studies here, starting in 74. And here I am looking about 12, but in actual fact, about 17 on the left there. I did have some inspiring teachers, and I just wanted to pick out a couple. The first one on the left, Susan Smith. Sadly, I couldn't find a photograph of her, although I did try quite hard. But she had legendary status among us as students simply by being the sister of Jim Clark, the racing driver. So that put her off to a good start with us, of course. But she was a veterinary pathologist, and she was the first person to say to me, "Do you realise you're actually good at this?" Actually, she's probably the only person that's ever said that. But it was hugely inspiring at the time, and she was a good role model as a female academic when there were very few female academics round about then, late 70s. And I think that did, although it took a while for me to get back to it, I think that did just stick in my mind a little bit that, you know, maybe this is something that I'm actually quite good at. The other person I'd like to mention is David Doxey, who some of you may remember. He retired about 10 years ago, I think now, but some of you will remember him. He's still very much alive. He was here last month, in actual fact. But he was a clinical pathologist almost before that was that term was used. And he really got me interested in clinical pathology as a student, and he subsequently became my PhD supervisor. So, in 1979, I started a PhD with him, and this was in the subject related to large animal clinical pathology, which I finished in 1983. And it had been my intention to go into large animal academic position, but there wasn't one. So instead, I was offered a position as a lowly demonstrator, really, in small animal medicine, again here at the Dick. And I thought, well, this is an opportunity. In those days, you just took what you could get, really, in a way. I thought, well, I'll just do this anyway. And it proved to be a hugely beneficial thing, which I think has stuck with me to this day, really, because having seen, it was a very busy position, having seen so many different clinical cases, it's helped me a lot in my current role as a clinical pathologist. And it also got me into the way of teaching vet students, too. And then, as luck would have it, and there has been a lot of luck along the way, I have to say, There was a position came up as a lecturer in large animal medicine here, again at the Dick. And I took that post and had it for the next, worked in that role for the next 10 years. At the time that I started, you just did horse and farm animal together, but about halfway through this time we were asked to specialise and I chose horses. There was a lot of clinical work in this, involved in this, and also a lot of night work, as my husband will testify at the time, but also teaching vet students, and there was an opportunity now to get into some research. So that's really the background, and I'd like to talk a little bit about some research that I did, and this is not the most scientific thing I've ever done, but I've kind of chosen things that I thought might be of general interest, and it really probably was the most satisfying bit of work that I've ever taken part in, because I think it did have some long-term effects on how this particular disease is viewed. So equine grass sickness, or equine dysautonomia, it's a disease still of unknown cause, and there is some association with Clostridium botulinum toxins, but certainly I wouldn't advocate that it's a cause and effect relationship. I think that it's much more complex than that, but there is certainly an association with Clostridium botulinum, the bacterium. And it causes degeneration of the nerve cells or neurons in the autonomic nervous system, which is the part of the nervous system that controls involuntary functions like heart rate and gut activity. And here you can see, this is a ganglion, which is like a nerve junction box from a horse with equine grass sickness. And at the top here is a normal neuron with that granular kind of appearance. All these pink ones here, these round pink things, are dead ones. So you can see what's happened there. Grass sickness occurs in two main forms, acute and chronic. And the acute ones are invariably fatal. They have to be euthanized, or to save them dying spontaneously, very severely affected. And I was much more interested really in the chronic cases. And in this type of grass sickness, it comes on much more slowly. There's severe weight loss, as you can see here, sweating, and other signs like muscle tremors. There's a lack of secretions in the nasal cavity, which gets very dry, and perhaps most important from the point of view of trying to monitor, manage these cases, difficulty eating and swallowing, and very poor gut motility. And at the time that I started working on this disease, the recovery rate for chronic cases was about 10%. And I can remember thinking to myself, if we could only just keep them going a bit longer, perhaps we might be able to get them to recover. So I decided that we'd undertake a drug trial, but in actual fact, this was only a small bit of what we were doing at the time. We were looking at all sorts of nutritional and management factors that would help improve the prognosis for these chronic cases. But just to explain a little bit about the drug trial, Cisapride is a drug that releases acetylcholine from the cholinergic nerves in between the muscle layers of the intestine. And it's supposed to increase coordinated peristaltic type activity, the propulsive activity of the gut. So it seemed like a likely candidate. And it had been used in horses for post-operative gut stasis previously. So it wasn't completely a shot in the dark, but not for grass sickness. And before starting the trial, you sit down and ask yourself some questions. Can we keep them alive long enough? Will there be any adverse effects? Can we withhold treatment if the drug is beneficial? And I mean, basically, what could possibly go wrong? Certainly the last one was very important, because anything to do with a trial like this, you can't withhold treatment if the drug is proven to be beneficial. Likewise, if there's some kind of problem that occurs with the horse and it can't complete the trial, you have to take it off the trial and continue with the management. These are people's animals. So, after some rather ludicrous trial and error, we opted for this method for monitoring gut motility. The very nicely named Precision Plastic Ball Company supplied these. So we gave these 6 mm pellets. 200 were administered by nasogastric tube or stomach tube on day 0 before we gave any of the Cisapride treatment. And we collected all the feces and recovered the balls over the ensuing week by, let's say, manual means would be the best to describe it. So we did that every day, and then on day 7 we started the treatment. And that was given every day for 7 days. And at the beginning of that, they were given another colour of plastic ball, so we could tell when they were coming through. Then we stopped the treatment after 7 days, and on day 14, after the treatment, we gave another colour of plastic ball and followed that through for a further 7 days, or at least until the balls had all come out. And this is just, this is not the whole amount of data, but I think it kind of gives a flavour of it. So down the side, we've got the groups. We actually had an injectable Cisapride group as well, but it gave very similar results, so I've not included that. But oral Cisapride, grass sickness affected animal group. Untreated grass sickness, and then healthy controls who only had the pellets given once. And you can see that there's a huge amount of variation. There's very large standard deviations here, but the healthy controls on average, it took 60 hours for half of the pellets to come through. So we've got the before, during, and after periods, but 60 hours for half of the pellets to be excreted. Before treatment on the grass sickness group that were treated, it was around about 146 hours. And you can see that after treatment, it was down to about half that. So transit time had halved after treatment. For the group that didn't get the Cisapride drug, they actually, for various reasons I won't go into, they started with a much better transit rate, and it actually got worse. And rather alarmingly, the mortality rate was actually higher in these horses as well. So you can see the numbers of horses are fairly small as a result of that, and as a result of trying to keep them going at the same time. So I think this drug did have some benefit, and this would be the first time that a drug had ever had any influence on the actual autonomic dysfunction in grass sickness, as opposed to just treating symptomatically, which was quite encouraging. Unfortunately, it's not available now. It's not used now for this purpose, at least. But I think it set this precedent that it was possible to influence the gut activity. The other aspect was the nursing side of things, and in the time that I was there, of course there was a team of us, it wasn't just down to me, we increased the survival rate of the selected chronic cases that we treated from around about 10% to 50%. And you can see this horse here, this is one of our early ones. This is him when he was the first affected, and this is him 4 years later. And it's really, a lot of it really is down to the nursing. So as I was alluding to before, I think this did have an effect on the way that veterinary surgeons in practice viewed this disease, that it wasn't necessarily hopeless. And quite a lot of these horses, in fact, in the initial study that we did, all of them went back to some kind of useful activity, sometimes even racing and eventing and that kind of thing. Although that's, you know, it's not always the case. So just to come up to the present day, this study is not finished yet, but it still intrigues me as to how some of these chronic cases managed to survive. So we were lucky enough to get around about 14 sets of tissues from horses that had had grass sickness between 2 and 18 years previously. Some of them were old friends, actually, from my clinical days. And we have this now have this unique set of tissues that we can work on. So some of the work I'm doing just now, which is not, this part of it is complete, but the whole thing's not. This is a ganglion from a control horse, and all these purple round things are neurons, which look normal. And this is one of the recovered cases that pretty much had completely recovered. And it was put down for something like lameness, I think it was. And you can see that there are hardly any neurons left, which is quite remarkable, really. Just to put some numbers on it, these boxes just give the range of values. So we've looked at 2 different nerve ganglia, different parts of the body, and this is grass sickness affected and normal horse, and the other ganglion, grass sickness affected and normal horse. And these are significantly different from each other in each case. Likewise, in the intestinal tract, this is the lower part of the small intestine, which is the worst affected part in grass sickness. I looked at two different nerve plexuses, one which is just under the lining of the gut and the other between the two muscle layers. And you can see that this is grass sickness recovered and normal, and the other one between the muscle layers, grass sickness affected, recovered and normal. And again, there's a huge difference between these. So certainly these horses, in spite of the fact that they had recovered, pretty much anyway, completely or partly, they do have significant depletion of the nerves within these sites. So we need to look for another explanation. And one possible explanation, or at least a partial reason for it, might be the role of the interstitial cells of Cahal. Cahal was he looks a bit like my father sitting here at his desk actually, but he was a Spanish scientist who discovered these cells back in the 19th century. And the interstitial cells of Cahal, or ICC as they're abbreviated to, these are pacemaker cells within the intestine. And they lie quite close to the neurons and the muscle cells and form connections between them. And this network seems to help in transmission of nerve impulses. Some of my colleagues at the DIC here, Neil Hudson, Joe Mayhew, who was here before, and Jeff Pearson, showed that there was a decrease in ICC in grass sickness, which seemed to be worse in acute than chronic cases. But they didn't have the opportunity to look at a number of recovered cases that we have the tissues for now. Unfortunately, I haven't quite finished this. I would have loved to have been able to give a lot of data on it, but I have looked at some of these, which have been quite recently stained up actually, and quite a lot of these horses have got intact networks of the interstitial cells of Cahal, which are these brown streaks there. So it's possible that these are maintaining the slow wave type activity, which is kind of a basic function of the gut, just slowly moving the food downwards, which is part of their role, even although the neurons are actually very badly affected. And if drugs could be developed to influence the interstitial cells of Cahal in horses, then there might be a way of supporting their gut function and perhaps improving the outlook further. So the next stage on the train journey, as shown at the top here, is I decided that a lot of my work that I'd done as a clinician actually involved quite a bit of pathology, and I was perhaps the words ringing in my ears, do you know that you're quite good at this? I thought to myself, I'll try and pursue this as a career. So in 1996, I moved to the Scottish Agricultural College, as it was called then. I hope though anybody from that organisation won't mind me calling it SAC. It's now SRUC, but it's a bit easier to say, I think. So they have a network of 8 veterinary laboratories across Scotland, and I went to the one down here in Dumfries in southwest Scotland. Any of you who don't know Dumfries and Galloway, this is a very beautiful part of the country. And instead of coming up the M6 and just keep carrying on to Edinburgh, you should turn left when you get to Gretna Green. Very beautiful place, and they have some very nice local native breeds as well, the belted Galloway there. Oops, sorry, going too fast. So I started there as a veterinary investigation officer, and then I was lucky enough to get the position of centre manager after the manager retired, and stayed there for the next 5 years. This was it was a very busy job, but very, very varied as well. And I learned a huge amount by taking that position, even although it was only for about 5.5 years. So the work that we did, well, and still goes on, of course, there, one of the major things is disease surveillance. And that took the form of post-mortem examinations and also laboratory diagnostics that we did in-house. And there were quite a number of other aspects to this job. Emerging disease investigation, so any unusual diseases or unusual manifestations of common diseases, we would go out and investigate, often on farm. Zoonosis investigation, so diseases that pass from animals to humans, occasionally the other way. And we also had health schemes for cattle and sheep, and I was in charge of about 1,000 of these flocks and herds. And that was really just to prove absence of disease in those flocks. The other thing that we did was support for government control schemes such as BSE, which was quite rampant at that time, and foot and mouth disease. And my time at Dumfries did coincide with the infamous 2001 foot and mouth disease outbreak. And some of you will remember that as well and are probably participating in it. That was a really horrible time for everybody. It was very harrowing, particularly to see the disease rampaging across my region, Dumfries and Galloway, taking out farms, people that we actually knew. It was very, very hard to see that. On a lighter note, we also supported the Scottish Marine Animal Stranding Scheme, which involved usually a phone call followed by a very long journey to some remote beach. If you were very lucky, you could find the beach, because that was pre-sat-nav. If you were even lucky, you could find the animal. And the tide hadn't come in the interim. And there was somebody there to give you a hand with it, because we had to manhandle these into the van, little Ford van thing that we had to get them back to the lab. On one occasion, we had a leatherback turtle, which is a couple of meters long. And the purpose was really just disease surveillance of these and toxin testing as well. There was one rather laughable incident when we were stopped by the police on the way back to the lab, and they had noticed that there was a dolphin's tail trailing along the A75, and they made us stop and cut it off and shut the door. But the anatomy, let alone the pathology of these animals, was absolutely fascinating. The adaptations for diving and so on. There wasn't much time for research at Dumfries, but during that time I did get interested in the parasite Neospora caninum, which is a protozoan. It causes central nervous system disease in dogs, and it's an important, perhaps more important than that, it's an important cause of abortion in cattle worldwide. And we certainly saw plenty of it in Dumfries. And some up-to-date figures are that it's the third commonest infectious cause of abortion in cattle in Scotland. Before you throw your hands up in horror at this, it's quite a complex life cycle, but basically there are two ways in which it can circulate, and the most important one is from the mother, and you'll probably see more of it in dairy cows than beef, although it does affect both, but you get a primary infection in the mother, and it goes vertically, by the vertical route, to the calf if she's pregnant. And this is the main way of maintaining infection within the herds. And of course, this grows up, is maintained in the herd, kept back, becomes an adult, and has calves, and the whole thing circulates in this way. There is another way, which is a minor way, but it may be one of the ways in which new infection can be introduced into herds, and that's via the dog as a definitive host. So the dog has oocysts that it passes in the feces, infects the cow, which eats whatever it is that the dog is contaminated, and then any placentas or abortion material may be eaten by the dog, and so it goes round here. And then this can go off in this direction afterwards. There are two main phases to this, or stages of this parasite. One is the tachyzoites, which are the rapidly multiplying form, and these multiply during kind of active infection, recrudescence of infection. And then the other one is the bradyzoite, which is more like a tissue cyst that's kind of walled off from the body's immune defenses, the bradyzoites. And most of the diagnostic tests that are available for Neospora caninum detect antibody against the tachyzoite stage. And that makes it very difficult to actually diagnose persistent infection, where the bradyzoites are mainly the ones that are present. which I'll come back to just shortly on the research side. So the research that I did at that time was to try to, and it was very kind of near market research, if you like, it was something quite applied, was to see whether measuring milk antibody might be a useful way of detecting infection. Mainly it's detected by microscopic lesions in aborted fetuses, and also serum antibody levels or blood antibody levels in the dam and the calf. But that's a very insensitive way, the blood is a very insensitive way of diagnosing it. And as I mentioned, mainly against the tachyzoite stage. So I was asking myself whether milk antibody might be useful, because it's much more applicable to farm situations. The farmer himself can, of course, collect the milk, and it might be useful for screening purposes. So a colleague, Mark Crawshaw at the AIR Centre, and myself embarked on this, as if the gas sickness wasn't enough of a punishment. We embarked on this rather ambitious study, which involved sampling 90 cows on 2 farms every month for a year. And it involved getting up at 5 o'clock in the morning and making a 130-mile round trip to sample these cows. However, it did show that there was a good correlation between the the serum antibody and the milk going up this way. There was quite a reasonable correlation. And the other thing we noticed, which had been seen before actually, was that they tend to fall into a low antibody group or a high antibody group. And there's a big kind of gap here in the middle. And these might be some of the persistently infected animals that are responding rather poorly to the tachyzoite stage. So that was really our conclusions from that, as I've just alluded to. And then again, coming right up to date, I was very lucky to be asked to be PhD supervisor of a PhD student, Stefano Guido, who was based at the Moore Dunn Research Institute, and he just finished actually. And his supervisors, they're Lee Innes and Frank Katzer. And I must say that they've done the hard work on this. So Stefano was looking at whether it might be possible to detect antibody against the bradyzoite stage. That's the tissue cysts that are so difficult to detect. And he found a couple of antigens that these animals, or some of them anyway, responded to, which was quite encouraging, really. The other thing that he did was to develop markers for genotyping Neospora caninum, which could have quite an important role in sourcing infection. And if you look at the antibody results that he obtained, screening a lot of cows, he found that using the tachyzoite test, the conventional test that's quite insensitive, 81.5% of the cows were detected as being positive. This is known positive cows, of course. And with the combination of the tachyzoite and the bradyzoite test together, almost 95% could be detected. So I think there's quite an improvement there in the detection rate, sensitivity, by combining those two tests. So hopefully he'll go on from there, study that a bit further. So the final stage on the journey was, I had the opportunity then to come back to the Dick, like I had a piece of elastic attached to me, got drawn back, and I was lucky to be offered the position of senior lecturer, this time in veterinary pathology, and that was in 2002. And I was made head of pathology in 2004. And as you can see, a personal chair, which is what we're here for this evening, 2008. So the inaugural lecture committee are extremely persistent, since that's almost 10 years. At that time, when I first came back, I didn't have any qualifications in pathology, so I set about rectifying that. And as was mentioned at the beginning, I'm now a fellow of the Royal College of Pathologists, diplomat of the European College of Veterinary Clinical Pathology, and a recognised specialist as well, which is easy to say in a sentence, but perhaps not quite so easy to achieve when working full-time as well. So one thing I thought might be entertaining to mention that I've been involved in since I came back to the Dick, with my colleague at the front here, Professor Anna Meredith, and we still do this together when we can, is red squirrel disease surveillance. This started as a hobby, actually, that Anna and I used to do in our spare time, but it quickly got out of hand as people started to send in lots and lots of squirrels. And who could resist, I mean, with such a beautiful animal? These come from members of the public, as well as from squirrel conservation groups and so on, Forestry Commission and so on. And we did have some rather laughable containers that they come in. Usually it's something kind of roughly squirrel-shaped, like a whiskey bottle container, carton, or Pringles we have here. This was a wrapping around a squirrel, looks like something from a murder scene. And I think my favourite one is this, which, in case you can't read it, ensure squirrel is fully thawed before reheating, which is from one of our regulars. But the reason that the surveillance was set up was firstly to just see what was out there in terms of disease that might be affecting red squirrels, and therefore, because they are an endangered species within the UK, and also to track the progress of squirrel pox, which is a fatal disease of red squirrels that's carried by the alien greys who are not affected by it. And they pass it on to the red squirrels and they actually die of it. And when we started back in 2005 doing this, squirrel pox was present in England, but it wasn't present in Scotland. And unfortunately, we were among the first to pick up squirrel pox as it came north into Scotland. And a kind of sad note, really, this is a very up-to-date map, which I only received this morning. And this little red dot next to Anglesey, it's actually on the mainland, is the first that has ever been reported in Wales, and that was about 2 months ago. It's a bit sad, really. That's progressing. Squirrel pox is a horrible disease. It causes skin scabbing, and as I say, it's ultimately fatal. And the way that we diagnose it is by electron microscopy, which is not your normal kind of go-to diagnostic test, but it's actually the quickest way to do it. And our colleague at APHA down in Weybridge, we just send them off and he reports it back on the day of receipt, which is remarkable, really. So just to show some data of what we've discovered on the many, now 500, this is 262 that I'm reporting here, but we've now done about 500 postmortems in total. And we had a vet student who took our blood-soaked scribbles, which were our postmortem reports, and turned them into something quite interesting. And this is only just a tiny bit of it, actually. So she worked out the distribution of disease. Of course, it's biased. If you're going to find a squirrel, you're going to find it squashed at the side of the road, most likely. So a lot of these are road accidents, but there were also some interesting other types of trauma, starvation, and squirrel pox itself, of course. And perhaps remarkably is this, most remarkable is this narrow grey bit which says "to decomposed," because it's really quite surprising how much you can get out of these carcasses. Hardly any of them were too decomposed to tell anything. But of course, any type of surveillance, unless you actually do something with the information, it's just like stamp collecting. So we discovered that there was a couple of interesting viral diseases that caused enteritis that hadn't been reported before, and also, perhaps most interestingly of all, this particular disease. And we'd seen these sporadically over the time that we'd done this study, started doing the surveillance. These squirrels have the swelling of the extremities, and you often see it most on the ears, the feet, the nose, and so on. And this proved, Anna Meredith and my colleague at the back there somewhere, Jorge Del Pozo, showed that this was actually a form of leprosy. So my original thought that this was nut allergy turned out to be incorrect, although it wasn't a terribly serious suggestion, actually. So I'd like to thank them for this data here. So what they did was to set about molecular identification of the mycobacteria from these cases of these swellings. And they found two related bacteria, Mycobacterium lepromatosis, which affected most of the ones that were found in the UK, and Ireland actually as well, and on Brownsea Island specifically, Mycobacterium leprae. And here's Brownsea Island down here. And it's quite intriguing, really, that Mycobacterium leprae was very similar to a strain found in a skeleton in Winchester, a human skeleton. which was 730 years old. So it does genuinely seem to be a type of leprosy of the sort that was seen in the medieval times, but in fact it's quite seen in third world countries to this day. So I think that was a really interesting thing that came out of the, our surveillance work that we were doing. So just to explain a little bit about what I do now, the role of the academic clinical pathologist. This was actually a little teaching session that I was doing with school students, actually, not vet students, but we do a very similar thing with the vet students. But the roles that I have, I work a lot of the time in the diagnostic service doing cytology, which is the study of fluid and cells and fluids, rather, for diagnostic purposes. Hematology, clinical chemistry, and other laboratory testing. I also teach. Resident training is a part of what I do too, research and the dreaded administration at the bottom. Cancers, of course, are very unpleasant things to have or to have in your animal, but they can look really very beautiful under the microscope, which is part of what I spend my days doing. Here's a bladder cancer. You can see this ginormous cell here, and an anal sac gland cancer, which has this rather nice rosette-like appearance. I also feel very passionately about the improving the training of residents who are specialising in veterinary clinical pathology. And I was fortunate to be in quite near the beginning of the establishment of the European College of Veterinary Clinical Pathology, and I've served in a number of capacities within that organisation. But it was one of the kind of younger of the specialties, and pretty much from a standing start, we managed to get, well, not us personally, but in the background we were organising and administering the development of the college, and now there are training establishments pretty much throughout Europe, and hopefully soon there will be going into Eastern Europe as well, and even there's one in South Africa too. So it's been a good experience to be involved in that. Finally, I'd like to talk just very briefly about the Centre for Comparative Pathology and give you an example of how that can work. So this was set up about 4 years ago or so, and the aim really was to integrate the study of human and animal disease, to gain knowledge about both and to benefit both sides. And this links the University of Edinburgh researchers in the University of Edinburgh College of Medicine and Veterinary Medicine and associated institutes as well, and elsewhere, not just within our university. And the idea was to integrate standard pathology techniques like histopathology, for example, with more molecular type techniques, molecular analysis, genomics, proteomics, and so on, in a wide range of species. And the director of this is my colleague, Professor Mark Ahrens, who's down at the front here too. And I must say it's been a pleasure to work with Mark. He's headed the Division of Pathology at the Western General. So just to give an example that I'm involved in myself, and there might be others in the room here who are also involved, because there's quite a number of us. So the study that we've undertaken is on canine primary secretory otitis media, which is a very long name, but some of you may know it possibly from your own experience or that of your children as a glue ear. And there's several of us involved. You can see myself, Michael Cheeseman from the Roslin Institute has been an important part of this, Tim Nuttall in dermatology in the Small Animal Hospital, Katya in neurology, Chiara, who's my colleague in veterinary pathology, Tobias in diagnostic imaging, and others from Roslin and QMRI as well. It's quite a multidisciplinary team. And I think it's nice that myself and Chiara, my colleague, have contributed to Michael Cheeseman's study of this condition in children, mice and rats, and he has contributed to our work in dogs. Here you can see, this is from Michael, this is the part of the middle ear of a mouse, and you can see that this fluid with the dark blue staining cells is built up within the middle ear. And the cause of this is not really very well understood, but It does seem to be something to do with the shape of the head of these animals, and the children seem to grow out of it as the head changes shape. And this has a bearing on the dog as well. It's much more common in the brachycephalic or short-nosed breeds of dog, and the Cavalier King Charles Spaniel is particularly predisposed. And here's a poor Cavalier with a head tilt here, obviously with vestibular disease, feeling dizzy, I should think. And you can see this material within, just bursting out through the tympanic membrane here, this glue-like material. On MRI scanning, this is the middle ear here, and it should be clear, full of air, and actually it's full of this glue-like material. And you can see it on histopathology as well from this post-mortem specimen. Here's the fluid. Here's the eustachian tube going from the pharynx to the middle ear. So just to show you a small snapshot, this is a much wider study than I'm showing the results of here, and it's still ongoing, but just as a kind of rough comparison as to what's going on, in our dogs that we've studied so far, the type of cells that are present within this fluid are mostly neutrophils and macrophages and fewer lymphocytes, and that's exactly the same as is seen in the children with glue ear as well. So we're hoping to go into this in a bit more detail. And we started, this has only just begun, but we've started to look with flow cytometry at what exactly these cells are, and we are starting to build up more of a picture of what, particularly what the lymphocytes specifically are, and then we'll work out what they're doing there. So what's my vision for the future of Easterbrook pathology where I work? I think the future looks molecular from where I'm, my perspective. My colleague Gavin Patterson, who was appointed last year, has improved, or introduced rather, quite a lot of kind of high-tech stuff, which we use in the diagnostic service, as well as having spin-offs for research as well. And he introduced the automated bacterial identification and antimicrobial sensitivity, the VITEC. Of course, it doesn't replace, and I'm sure never will really. There's always going to be some place for bacterial culture, which of course we do as well. And Gavin's also introduced ribosomal PCR and sequencing, which has spun out to the diagnostic service, and he's managed to find some organisms, both fungal and bacterial, that we hadn't recorded before. I'd also like to develop flow cytometry for leukemia diagnosis in our patients here, which I hope will be done in the next 12 months. So finally, just to round up, I'd like to make some acknowledgements. Funding bodies, of course, this is not exclusive. I've had many more than this, but these are the people who funded the work that I've talked about this evening. And particularly, I'd like to mention the Equine Grass Sickness Fund, who've actually funded, it's just a small charity, they funded all the grass sickness work that I've done over however many years it is, many years. I'd like to acknowledge Professor Sir John Saville and David Argyle, our head of school, who's not here today, but they appointed me to my current position. I would like to thank them for that, and also for their support for the pathology as a subject and for our team. Very few things that I've done have been done in complete isolation, in fact, probably nothing. So I'd like to also acknowledge colleagues past and present and the teams that I've worked in, and probably the best team I've ever been a member of is the Easterbrook pathology team, who are a group of hard-working individuals who, a very cohesive team who work very, very well together. Just to embarrass her completely, I'd like to mention Shona Smith, Dr. Shona Smith, who is head of anatomic pathology and is my deputy, if you like. She puts in a pile of work behind the scenes that people don't always notice, apart from me. I'd like to dedicate this lecture to my family, to my brother, and long-suffering husband of 34 years. 34 years married, not 34 years of age. But they've put up with me through thick and thin, and they're here in person. And two people, my parents, who are here, I'd like to think in spirit. Thank you very much.
Elspeth, thank you. That certainly was a romp around being a veterinary scientist. I think it's really quite rare to have a talk that actually covers so many different aspects, and to be able to, in the same talk, to reference Cahal, to reference Bob Edwards, but also slip in Jim Clark, I think is really quite an achievement. The range of animals and diseases that you discussed, going from leatherback turtles to red squirrels, to dolphins, through to BSC and foot and mouth disease, cancers and glue ear. But I guess in a way there were three images that have really stuck in my mind. The first one was the use of the plastic balls by the Plastic Ball Company to measure gut transit. And I was, especially because I'm colorblind, and I was especially thinking, how would I do that with those red and green colored balls in the mess that you'd have to do? That was the first thing. The second image that really stuck with me was the dolphin tail sticking out the back of the van going down the road. And I think probably the one that really stuck home the most was how vets have to deal with many varied people, individuals and circumstances. And I think the squirrel containers were my favourite of the lot. So Elspeth, thank you. That was a wonderful evocation of life as a vet. We maybe had to wait 9.5 years, but I think everybody will agree it was worth the wait. Thank you very much.